We're going to be talking about massively parallel sequencing, which you may also hear referred to as next-generation sequencing. The objectives of this lecture are to become familiar with the general theory of how massively parallel sequencing works, and then also to become aware of some of the pre-analytical, analytical, and post-analytical metrics that you want to pay attention to help guide the interpretation of results from these platforms. To begin with, we should talk about the difference between traditional Sanger sequencing versus next-generation or massively parallel sequencing. Sanger sequencing starts with the amplification of a single amplicon of interest, and the millions of copies that are generated through polymerase chain reaction are all sequenced at the same time. In that setting, the Sanger sequencing is highly precise. However, the molecule that is most abundant in the mixture that you made during your polymerase chain reaction is the molecule that you'll be sequencing, and therefore the sensitivity of that platform is somewhat limited. And indeed, anything that is present at less than 20% of the amplicons that you make is not reliably detectable through Sanger sequencing. Next generation or massively parallel sequencing, on the other hand, begins instead with the generation of a library, and we'll talk a little bit more about that in a few slides. And because of the complexity of it, you actually sequence every individual molecule of that library individually. So as you can imagine, it is a much more flexible platform. It can be used for much broader applications, and we'll talk in a few slides about how it actually works. In the context of clinical next-generation or massively parallel sequencing, there are really two platforms that you will hear referred to continuously. One of them is the Illumina set of sequencers that include the MiniSeq, the MiSeq, the NextSeq, the HiSeq, and the HiSeq 10. And on the Thermo Fisher side, the PGM, or the Personal Genome Machine, the S5, and the Proton. And these are different chemistries, but they're the two platforms that you will see most often in the clinical setting. But despite which platform you may use in the clinical setting, there is really a sort of generic workflow for how massively parallel sequencing works, and it's depicted in this slide. And so for every massively parallel sequencing assay, you essentially have three steps. There's library preparation, clonal amplification, and then followed by sequencing. The library preparation is a key step in what makes massively parallel sequencing so useful and so flexible. And so rather than doing a single PCR of an amplicon, such as you would do for Sanger sequencing, the library preparation allows you to prepare fragments of all of the areas of the genome that you might be interested in. So it may be all the coding regions of a specific gene, it may be the coding regions of multiple genes, it may be all coding regions, or it may be entire genomes. It's an extremely flexible thing. And to make your library, you can either do it as a PCR-based amplification approach using primers generated to specific areas of the genome, or you can do it as a hybridization by generating probes that then select and hybridize to fragments of genomic DNA. In either case, at the end of the library preparation stage, what you have is a complex mixture of all of the different areas of the genome that you are interested in. That mixture then goes through a small clonal amplification, and that's just to bump the signal up. But the important thing is that prior to the clonal amplification, you actually separate every fragment of the library that you made into a separate reaction. So you're essentially sequencing every fragment in that originally library preparation on its own. And so when you have completed the sequencing phase after the clonal amplification, you can essentially build back through bioinformatics and alignment against a reference sequence, you can build back the complex mixture of everything that you've sequenced. And as you might imagine, you can get a highly sensitive representation of all of the different fragments that are in there. So in the setting of somatic cancer, if you happen to have a variant that is present at less than 20%, you actually will be able to see it with massively parallel sequencing because the fragments containing that variant are actually individually sequenced away from the rest of the fragments. Here's a little bit of a blowout then. You see the library prep, the sequencing, and then the bioinformatic analysis across the top. And then the bioinformatic analysis is blown out a little bit in section B below. And what you can see is that the analysis of next-generation sequencing is really quite a complicated process. You start off with the raw reads, so those are the reads from each of the individual molecules that you made in your library. The demultiplexing, mapping of the reads, on-target filtering, quality filtering, assigning or assessing the depth of coverage, and getting to the point where you have ready reads for analysis is something that's generally done by bioinformatic pipelines that are embedded in the platforms that we're using, but it's important to be aware of them. And it's also important to realize that there are statistical metrics at each of those stages that allow you to remove reads that are not of a quality that you want to include in your final clinical analysis. Once you've gotten your ready reads for analysis, that's when you can actually make the calls of the variants which are seen as differences from a reference sequence. And once you've made the calls, then you can actually do the clinical interpretation. So there's a fair bit of work that has to be done downstream of the sequencing in the setting of massively parallel sequencing. Massively parallel sequencing, by everything I've just described, I think you can appreciate is a highly flexible platform. And so, as I've already mentioned, it can be used to look at everything from the whole genome to just the exome, to just amplicons of interest. You can also use it to look at RNA. You can also use it to look at methylation patterns. And it's basically as flexible as your imagination. In the setting of the clinical assay, most people at this point in time are using what we would call targeted assays usually for hotspot areas in key genes that are known to be important in whatever the condition is. Regardless of what you're using, though, for your MPS assay, you need to go through the steps of clinical validation. And this really isn't specific to NGS. However, it is a little bit involved when you're doing an MPS assay validation. Think about the clinical validation as dividing into three major areas. There's the test development, then there's the test validation, and then there's the ongoing quality management of the assay. Test development is where you optimize your components and your test in general. So you're looking there at the general assay conditions, what kind of coverage do you need, which amplicons do you want to cover, how many samples are you going to be able to put into a batch, what kind of analysis setting and thresholds do you want, and quite often a lot of this is taken care of for you if you're buying a ready-made panel. However, if you're making your own custom panel, you have to think about all of these things. This is not actually part of test validation. This is the development of the test, and only once you have optimized all of those parts of the test do you move into validation. When you're validating, you have actually locked down how you're doing the assay and how you're analyzing the assay, and now what you're doing is making sure that the assay works and reaches the required sensitivity, specificity, robustness, and reproducibility for any sample type that you hope to use in the clinical laboratory. You also want to validate for every different type of variant that you might want to look at. And so massively parallel sequencing, of course, looks at different types of variants, which include single nucleotide substitutions or variants, small insertions or deletions, but it can also look for fusions if you start with RNA as the input material, and it can also follow and track copy number variations. For any of these different types of variants that you might want to be looking at, you need to make sure that your platform has been validated for that general category of variant. And then, of course, as for all clinical assays, there's ongoing management and quality improvement, which includes making sure that you have external quality assurance assays that you're doing regularly in the laboratory to make sure that your platform is performing as you think it should. So when you think about massively parallel sequencing in the context of pre-analytic, analytic, and post-analytic metrics that we might be interested in, here are some of the sorts of questions that we would be thinking about that I've listed on the side of this slide. And I'm not going to read through all of them. And again, most of them are not particularly specific to MPS, but the considerations actually are really important when you're thinking about something as complex as an MPS assay. So if we start off by thinking about some of the pre-analytical pitfalls, and I'm not going to have time to go through every single one of these, but if you look at the pre-analytical pitfalls, here's a table from a paper that was published in 2014. And this paper, or this table, has always struck me as being somewhat surprising that as late as 2014, and I actually don't think things have changed too much in 2018, this is looking at all of the pre-analytical factors in the development of an FFPE, or a formalin fixed paraffin embedded specimen, which is really the specimen type we use most often in the context of cancer, at least. Each of the pre-analytical factors and how well they have been investigated in terms of their impact on the quality of DNA, RNA, protein, and morphology. And what you can see is that on the far left of the slide, there's a couple of these factors that have been analyzed comprehensively for all 4 of the analytes. In the middle, you'll see that there are some that have been analyzed for some. And on the right-hand side, you'll see that there's a lot of things that still have not been looked at all in terms of their impact. And so the take-home message from this is that although we recognize that the sample and its processing and everything about it is really part of the pre-analytical parameters that we need to think about, many of these parameters are things that we really never get a chance to look at, but they may actually impact the quality of how your assay performs. And you should at the very least be aware of them, even if you can't control them all. One of the very important pre-analytical steps in the assessment of at least somatic cancer is estimating the percent of tumor cells that are present in any given slide. And this is important because it actually speaks to the sensitivity of the massively parallel assay and whether or not you expect that you should be able to see any variance depending on whether or not there are sufficient tumor cells present. And so it does actually make sense to think a little bit about what are we asking people to do. So when we ask people to estimate the tumor percentage in an area that has been selected and submitted for molecular analyses, if you look at a slide like this, one might argue there are roughly 100 tumor nuclei and maybe 200 normal nuclei in this little image. And so you might say, a pathologist might tell you there's about 33% tumor cellularity. However, it's important to note that this sort of estimation, the eyeball estimation, is often not going to be particularly accurate, not through any fault of the pathologist, but just by the fact that tumors are not usually diploid. Tumor cells, I mean, are not usually diploid, and therefore you can see the calculations as what happens if that tumor cell is not diploid in terms of our estimation of how much of the DNA is actually coming from a tumor. It's also true that tumor nuclei tend to be different sizes compared to normal, and that does lead sometimes to a misrepresentation of the percent of tumors. So really, and the other question is how close the H&E slide from which the estimation is being made are to the sections that are actually received in the molecular lab. This all matters because we're relying on the fact that there are a certain number of tumor cells present in the material that we're scraping off the slides in order to make an assessment of whether or not we're going to be able to see a variant. And given the fact that massively parallel sequencing is so sensitive, it's actually quite dangerous in some ways then to get a negative report from somewhere where you believe that there were sufficient tumor cells for assessment, and yet in fact the estimation of tumor cells may have been incorrect, and the negative report may actually in fact be an inconclusive report. In terms of analytical pitfalls, there are many that we could talk about. Certainly the quality of the library that you generate, whether you generate it by a PCR or by a hybridization approach. You may get poor coverage of certain regions. You may get poor distribution of the fragments of sizes of DNA before you even start making your library. You may get very uneven coverage, which isn't really a problem if you're just looking for single nucleotide variants, but does become an issue if you also think that your assay is going to uncover copy number variants for you. If you have non-uniform depth of sequencing across fragments, that is an issue for copy number calling. If you have non-uniform quality of bases, and we know that that happens especially as you read towards the end of every fragment, the quality of the sequencing drops off. And so it's important that you actually remove the poor quality bases and fragments from anything that you're looking at. You may have what's called a strand bias, and a strand bias is where you see a variant if you're looking in one direction of reads, but if you sequence in the other direction, you don't see that variant. And so trying to sort out which of the calls is actually the right one becomes important, and it's something you need to look at. And of course, the reach homopolymer runs are sort of infamous regions. A homopolymer run is somewhere where you have the same nucleotide repeated more than once, and some of the platforms have difficulty in really assessing the true number of nucleotides in a homopolymer run and can actually make mistakes, especially when it comes to insertions or deletions in these areas, because there can be easily misalignments. So all of these things are things to be aware of. The easiest way to make sure that you're actually looking at a lot of these variants, other than just looking at the QC metrics that are generated every time you do a run, is to take a look at the pileups of the reads. So this pileup diagram is really what has replaced what used to be the sort of nice chromatograms that we saw in Sanger sequencing. Now instead we look at these pileup runs, and you can see here the forward and the reverse strand coverage. You can see the runs depicted as bars underneath that sort of coverage plot, and you can see where the mismatches against the reference sequence are. And when you look at these pileups of the reads, what you get a good sense of is the depth of coverage, the uniformity of coverage, and you also get to look at the mismatches against the reference sequence and try to assess whether or not you think they are real. And clues about whether they are real or not would be, for instance, if you saw a variant, a mismatch, but it only occurred in one particular amplicon and not in other amplicons that overlap the same region, or if you saw a variant that only occurred in amplicons reading in one direction, so that's the strand bias that I mentioned, those are all clues that perhaps you are looking at something that is an artifact of sequencing. You can also look at the quality metrics, as I mentioned, and you'll get that from the FASTQ file that you generate every time you run an MPS assay. And here's just an example. Here we're actually looking at the quality score and the base calling accuracy. And so the quality score, which is given as a Q number, and you can see down here Q10 refers to the probability of an incorrect base call of 1 in 10. Q30 is 1 in 1,000, and so we're often aiming for at least a quality of Q30. And if you look at these two little charts up here, you're looking at on the x-axis, it's the distance from the beginning of the read. On the y-axis is the quality score, and you can see that good data has high quality across most of the read, but even good data, the quality does begin to drop off as you move towards the end of the read. And then on the left, you see the bad quality data, where you see that the quality score just tanks very early in the read, and this would be a read that you'd actually would like to discard. Most platforms, the Illumina and the Thermo platforms, and most assays actually have default quality control metrics that you should be paying attention to. However, you can't just use sort of out-of-the-box metrics. You need to ascertain that the metrics work for the assay in your lab and that they actually support what you say your assay can do in terms of sensitivity and specificity. So you need to ascertain them, you need to document them, and then you need to use them at the sample level and at the gene level and even at the nucleotide level. So you want to make sure that you have metrics that are specific to every kind of variant that you want to identify and for each sample type that you're going to use, and then again, of course, for each assay that you run. One important metric is something called limit of detection, and the limit of detection in some ways is really the sensitivity of the assay. It really has to do with how many fragments with a mutation need to be present as a proportion in order for you to reliably identify that variant. And the limit of detection is something that you can actually manipulate to some extent simply by sequencing more depth or more often through a specific region. One important question becomes really the question of your limit of detection or your sensitivity versus the biology of what's actually going on. And I think this is where, as lab directors who are making clinical judgments about the impact of a variant, it's kind of important to have this in your head. So often, and certainly in our case, we may have a limit of detection of, let's say, 5%. So what we're saying is that if a variant is present at 5% of the molecules that we're sequencing, then we will reliably be able to tell the clinician that the variant is there. So if you look at the little cell on the right-hand side that I've depicted, you can see that of the cells that are there, the tumor cells are the red stars. You can see that 10% of the cells in this little section are tumor cells. And in this case, imagine that 100% of the tumor cells are heterozygous for a mutation that you're interested in. And if you do the math, you can say, well, the variant is present at 5%. And we would be able to detect that in a massively parallel sequencing assay that has been validated to a limit of detection of 5%. But if you look at the cell, or you look at the section on the right-hand side of the slide, in this case we have 25% tumor cells. 40% of those cells are heterozygous for the mutation of interest, and the variant is present at 5%. So we will also be able to detect the variant of the massively parallel sequencing assay, validated to a limited detection of 5%. But biologically, that's a very different situation. Because on the one hand, on the left-hand side example, all of the tumor cells essentially have the mutation, and one might expect that if you are detecting a variant that is a target for a therapy, that patient might do quite well on the therapy. Whereas on the other hand, even though it's still 5% and we're still detecting it by our assay, The fact is there are tumor cells there that are not carrying the variant of interest, and therefore one might expect that the clinical outcome for that patient would be quite different. And I think it's important to recognize that the technical parameters have actually the biological parameters, have an impact on the interpretation, even when all technical parameters appear to be met. In terms of post-analytical considerations, there's a couple of important questions. Are your quality metrics established? Did your assay meet those, as we've been saying all along? Your analysis pipeline also needs to be well documented, needs to be validated, and needs to be up to date. And when I say up to date, that includes every single time a piece of software is upgraded from the vendor, you need to actually re-verify or sometimes even revalidate your assay to make sure that you're still calling variants the way you think that you are. And of course, it's going to be important to recognize the difference between variant calling and variant interpretation. So this is really a combination of both of these things, and I showed you. So when it comes to variant calling, there are some pretty key metrics such as the depth of sequencing that I've already spoken about, checking the quality metrics like the quality scores that I showed you. And then also it's important to check the context of the variant. And I've already mentioned homopolymer tracks to you. And the best advice I guess I can give is that you should be using a visualization tool such as IGV, and here is a screenshot from an IGV, basically to sort of help you decide whether a variant that is being called is real. And so here's an example. You can see the reference sequence across the top in color here, and then you can see the reads are aligned in the shaded gray bars here. The arrow on the end of each of these reads designates the direction in which the sequencing was being done. And what you can see here is that there's a call that basically says there's a C to T that has been replaced by a TC. And so this would be a mutation. But if you look closely, what you'll see is that that only seems to occur in one set of amplicons that run in one single direction. You don't ever seem to see it in different amplicons that cover the same region, and you don't seem to see it going in the other direction. So this would be an example of a potential artifact, a strand bias artifact actually. It only happens in the one direction, and it only happens in one kind of amplicon. And so you would probably want to go in and look at this again in a second sample to make sure that this is actually real, which in this case it probably is not. Overall, I think the most important take-home message is that you have to understand the limitations of the platform that you're using, as always. And in the context of massively parallel sequencing, this can be somewhat complicated. So you need to understand the limitations of the software in the pipeline that you're using. You need to understand the limitations of the platform that you have chosen for that specific assay in your lab. You need to understand the limitations of the assay itself. And you need to understand the limitations of your sample and what that does to your ability to interpret variants. And so I just in the last few minutes want to show an example of each of these sorts of limitations and pitfalls that you might want to think about when you're doing MPS in the lab. So understand the limitations of your software, and unfortunately this doesn't capture particularly well, but here is an example of an assay. This was actually a melanoma sample, and what you can't see easily, but here I'll just bring it up on the side, is that there were 2 BRAF mutations that were called in this sample, and that's what these lines are here, and I've just brought them up here on the side. So one of them was the C1799G to A, which would be a valine at 600 to a MET. And on the other hand, there was a valine to a lysine, which is actually a deletion of the GT and an insertion of an AA. And both of these were called at essentially the same variant allele frequency and with the same quality metrics, suggesting that the software was very convinced that both of these variants were present. So you have to ask yourself, how likely is it that both of these variants were present in a single melanoma sample? And if it is this deletion of the deletion of the GT and insertion of an AA, but you never actually had this situation. And what's happened in this case is that the software has actually decided that it reads along until it gets to this first change, and then it makes the call that that is a valine to MET, and then it reads along and sees the second change and makes the call that it's a valine to lysine change. But in fact, the first call is erroneous. And this is really just the software that is trying to make decisions about what it's seeing in the alignment. And again, although in this case it actually wouldn't make any difference for the patient because it's still a V600 mutation and the patient would still respond presumably and be treated the same way with a BRAF inhibitor, one could imagine that if this happens in a different kind of a gene, that might actually be important. And the fact of the matter is, the valine to MET call is actually incorrect. So here's an example where you need to know about the limitation that the software tends to do this sort of thing, and you need to check these calls by looking in IGV. Understand the limitations of your platform. So here is an example. This is actually from a publication. This is not from our lab in particular, but what this publication showed is this is a lung tumor, and this is a sequencing of a region of EGFR. And what you can see is that there are several calls here in this IGV trace. And so they have basically blown up this piece of the trace down below. And what you can see is that if you just look at the EGFR part, you can see that there are a number of different amplicons covering this region, but you can see that these calls here, these 3 calls, are only occurring within this one specific amplicon. And it turns out that this is actually an artifact of the platform itself. This is an assay that the amplicons are generated by PCR, and it turns out that the primers that are made to that are generated to make this particular amplicon can actually bind in more than one place. And so you actually have erroneous binding of the primers in another place in EGFR, which generates this fake mutation over here. Sorry, I can't make my pointer work here. Anyway, so you have this erroneous binding of the primer in a different place in EGFR, but because it isn't binding with the 100% overlap, it actually generates a mutation because of the sequence of the primer, and that mutation is then seen only in that specific amplicon. And if you look at the correspondence between the ability to see that versus the quality of the DNA, it turns out that in DNA that has very low quality, you see a lot more of that kind of mispriming. And so here's an example of where your platform actually has a limitation. The assay has a limitation and the sample has a limitation that may actually introduce an erroneous call simply because of mispriming. Now, this is something that quite often when this happens in an assay, the assay is flagged, and this becomes what's known as a black box call or a blacklist call. And so, you need to be aware of these blacklist calls. But it is something to understand that sometimes if you're generating your amplicons through PCR, that you can have mispriming and various other events that can cause incorrect calls. And again, the clue about whether or not it's an incorrect call really comes from looking at that IGV trace, where you see that this mutation is present in multiple amplicons, but these 2 mutations are actually not, and therefore they are not real. Understand the limitations of your assay. So here is an example. This is from a long QT run that we were doing. We were looking at mutations or looking for mutations in the KCNH2 gene. When we sequenced the gene for this particular patient, it was completely wild type, so there were no variants found in KCNH2. However, because we are aware that KCNH2 often has mutations that are not single nucleotide changes or small indels, they're actually major rearrangements of the gene, we also analyze our NGS run, in this case looking for copy number variants. And when we did that by simply looking at the consistency of amplicon calls across the KCNH2 gene, we saw that there was a big dropout of a particular region, and so we understood that there was a deletion, and this actually corresponded to a piece of exon 3, and so we were able to say, oh, well, wait a minute, it's not a wild-type KCNH2 for this patient, there is actually a major deletion in exon 3, so that's the sort of first step, which is you need to know that your assay, you need to look at your assay in different ways to be able to find the exon 3 deletion, because you wouldn't see that simply by looking at the sequencing traces. You actually have to look at the depth, uniformity of depth across the gene. So that would be the first key. However, when we actually looked at the split reads, and these are reads that are actually split because of this, these are reads that go over the breakpoints of this deletion, and they're what's called split reads, because part of the read is actually reading correctly, and then the other part of the read is kind of dangling in the wind because of this deletion on some of the molecules. When we looked at those split reads and tried to make sure that we could see the breakpoints, they didn't make a lot of sense. And it ended up being that the actual mutation in this patient was quite a bit more complex than an exon 3 deletion. And I've tried to show it here. It actually ended up being a deletion of a piece of exon 3 followed by 2 separate inversion events, which resulted basically in scrambling the entire KCNH2 coding region. Now, again, in terms of the clinical interpretation for this patient, it would have been enough for us simply to find the exon 3 deletion and report it that way, because that would be enough to say that the KCNH2 protein is probably non-functional, and therefore that's a pathogenic mutation. However, if you actually want to get the appropriately correct call, especially if you want to start looking at this segregating in other individuals and possibly having other labs looking at other family members, it is important actually that you get the call more accurately correct, and therefore you have to understand, again, your assay doesn't necessarily tell you right away when these kinds of complex events are happening. And then finally, we want to understand the limitations of the sample. And so here's an example of a breast cancer specimen. And when we ran a panel, which is called, it's a comprehensive panel, it has about 130 some genes on it. And we found 94 different mutations in 45 genes that were detected by this targeted sequencing platform. And you have to ask yourself, is this a tumor that has a very high mutation burden? And of course, we're all very interested these days in tumor mutation burden, and therefore, this would be something you might flag as being a sample that has a really, really high mutation burden, and therefore that would be clinically important. However, when you go in and look at the metrics of this sample, you could see that only about 50% of the reads mapped on target. The average depth was extremely low at 61. And actually, normally we would want these at about 400. The uniformity of coverage, which is normally at about 98 to 99%, was only about 57%, and the mean read length, which should have been closer to 200, was only at about 66 base pairs. And so what you can see is that what looks like a really high tumor mutation burden actually translates into a very poor quality sample. And we've seen this repeatedly, that the quality of the samples really impacts the output of the reads. And so you need to be aware of everything from the very beginning of when you first get the sample in the lab to the very end where you're making the clinical calls, that each of the steps in the process along the way impacts how you're actually going to use these assays, how you're going to make the interpretations, and what you are going to say to the patient and the clinician when you use these assays in the clinical setting. Thank you for your attention.